All right, we're going to end this unit on mitosis and meiosis by kind of looking at how they are different. So we already know that mitosis makes two cells, and each of those cells are identical to each other. And meiosis does not make two, it makes four cells. So meiosis makes four, and they are all genetically different. So we have two differences there, 2 versus 4, identical versus different. And then if we take a look at the number of chromosomes that they contain, we find another difference. Mitosis is 2N, a diploid, 2 sets of chromosomes, where meiosis is just N, or haploid, and only contains 1 set of chromosomes. Mitosis is used by cells that reproduce asexually, like the hydra, where only one parent was needed, whereas meiosis is used by organisms that need 2 sexual partners in order to reproduce. Visually, I want you to take a look that they do have some similarities. So we have, for instance, your diploid parent cell, and this is replication, so that you can see all we've done is we've created kind of that identical copy, and they're being held together, each one by the centromere, holding the 2 halves together. And at the end of mitosis, we still have identical to our original cell. Each one is identical to the parent. Whereas in meiosis, we have those 4 chromosomes there, but in each one of our cells we now have 2. So they're definitely not identical to the parent. And then we also go through yet another conversion, another cut. And now instead of the sister chromatids being attached, now they're not. So now we have the 4 different cells at the bottom for meiosis. And now we need to look at differentiation. I'll say that again because it's one people are always like, how do you say that? Differentiation. All right, and this is where a zygote, which remember is a fertilized egg, goes through the process to become specialized. You and I all started out the same way. We all started from a sperm and an egg cell that combined, and then from there we have you and I. So we have a brain, and we have a heart, and we have a stomach, and we have skin cells, and we did not have any of those things when we first started. So how in the world did a single cell zygote right here, single cell, end up creating red blood cells and white blood cells and skin cells, and how did we create all of those things? And that is called differentiation. This is where a single cell, there's a whole bunch more biology we could get into about this, but at some point during your development, while you were still, in the uterus being developed, you went through this differentiation process, and each one of these layers designed and can start to create all of these different things: skin cells, neurons, the pigments that you have, cardiac muscle, which is your heart, red blood cells, cells in your kidney, we have pancreatic cells and thyroid cells. And so all of that happens through this zygote being fertilized. So that is, just huge, that we can start out as a single cell and then become trillions of cells that are so unique. I just find that amazing. Moving on though, as far as what increases the genetic variety between us and why you might look different but have some similarities to your siblings, one of them is independent assortment. So this is when your mom and dad chromosomes get separated from each other, during anaphase 1 of meiosis. And independent assortment, again, remember we had this picture earlier, it's just a matter of the way they line up as to how they're going to get broken into the 2 cells. But each gamete has approximately 8 million different combinations. So 8 million different egg possibilities, 8 million different sperm possibilities. That's a lot of variety there. We also have the crossing over. The crossing over again is where your paternal and maternal, the homologous chromosomes are going to trade pieces, and that just provides new combinations. So if this particular, if the dad's chromosome was the one that gets passed on, it's not fully dad's chromosome anymore. It has a little bit of information from mom, but not all of it. So again, there's just different combinations there, and this is a very important source of genetic variety, variation among siblings and among family members. And then lastly, we have random fertilization. So fertilization, when that zygote, the sperm and the egg come together, we know that we have 8 million different egg combinations, 8 million different sperm combinations. That gives us 64 trillion possible diploid combinations. So your parents could have 100 children, and they would still all be very genetically different. Some families, siblings look more like each other than in others, and that's just kind of the way it works. You know, there are a lot of sperm cells swimming toward that egg, and it is 1 out of 8 million that could possibly reach that egg. And so the chances of any two siblings being identical, except for identical twins, you know, you have a 1 in 64 trillion chance of that. It's just incredible the amount of variety that exists because of independent assortment and crossing over.